Welcome back to the deep dive. Today we're looking at a group of organisms that essentially define the concept of punching above your weight class. We are talking about the Picornaviridae.
Yeah, it's a perfect description, really. The name itself is this great mix of Latin and modern biology. Pico, meaning small, and, well, RNA, describing the genome. These are the small giants of virology.
And when we say giants, we aren't talking about physical size. I mean, they are actually among the smallest viruses we know, but we are talking about impact.
Right.
This is a family that spans the entire spectrum of human misery. From the annoying sniffles of the common cold to the devastating, life-altering paralysis of polio.
Exactly. And for you medical students or professionals listening, this is an absolute high-yield minefield. We are basing our deep dive today primarily on Murray's Medical Microbiology, Chapter 46.
With a little bit of JWET's Chapter 36 sprinkled in for some structural context.
Right. We are going to deconstruct this family, specifically focusing on the enteroviruses and rhinoviruses. Because you really need to understand how two viruses with such similar genetic blueprints can cause such radically different diseases.
That's the central mystery we want to solve today. How does one cousin destroy your motor neurons while the other just, makes you sneeze? Let's start with the basics of classification. Murray lists a huge number of genera in this family. It feels a bit overwhelming.
It definitely can be. There are over 230 members in the Picornaviridae family. But for clinical medicine, you really only care about 5 main groups.
Okay.
You have the enteroviruses, rhinoviruses, hepatovirus, which is hepatitis A cardiovirus, and aphthovirus.
But for today, we are zooming in on the two that cause the most confusion and the most diverse pathology, which are enterovirus and rhinovirus.
Right. And while they share a lot of genetics, they have one fundamental physiological difference that dictates everything about their clinical presentation.
This is that tank versus drone concept we discussed in prep.
Yeah, it's a highly useful mental model. Let's look at the enteroviruses first. These are the tanks. They are transmitted primarily by the fecal-oral route. Now, think about what that entails. To successfully infect a host via that route, what does a virus have to survive?
Well, it has to survive the stomach. It has to survive a pH of 3 or even lower.
Exactly. And enteroviruses are acid stable. You can literally drop them in a beaker of acid, and they are fine.
Wow.
They are designed to pass through the gastric bath, enter the intestines, and replicate there. They're armored against the environment. They resist sewage treatment, heat, and detergents.
Contrast that with the rhinoviruses.
Rhinoviruses are the drones. They're specialized for a very specific environment, which is the upper respiratory tract. They are acid-labile.
Meaning if you swallow a rhinovirus, your stomach acid destroys it immediately.
It's gone. It cannot infect the gut because it cannot survive the journey.
And there is a temperature component here too, right?
There is. Enteroviruses are comfortable at body temperature, so 37 degrees Celsius.
Yeah.
Rhinoviruses, however, have evolved to replicate optimally at 33 degrees Celsius.
Which just happens to be the ambient temperature of the nasal passages.
Precisely. The nose is cooler than the lungs or the gut because of the constant airflow. So rhinoviruses are restricted to the nose and throat, not just by receptors, but by their sheer inability to handle heat and acid.
That one distinction, acid stability versus acid lability, explains why you get diarrhea or systemic disease with one and just a runny nose with the other.
Exactly.
Okay, so we have the classification anchored. Let's move to the physical architecture. What does a picovirus actually look like under the microscope?
Imagine a microscopic soccer ball. It is an icosahedral capsid, which is a geometric shape with 20 faces. And the critical feature here is that it is a naked capsid.
Meaning it has no lipid envelope.
Correct. And this brings us back to the durability we just talked about. Enveloped viruses like influenza or SARS-CoV-2, they rely on that oily lipid membrane.
Right.
If you dry them out or hit them with soap or alcohol, that membrane pops and the virus dies. But a naked capsid is just a dense protein shell. It is tough. This is why enteroviruses can survive on door handles or in swimming pools for days.
Exactly.
So let's drill down into the proteins that make up that shell. The text mentions VP1, VP2, VP3, and VP4.
Right. The capsid is built from these 4 building blocks. VP1, VP2, and VP3 form the outer surface of the shell. They are the actual armor plating. But VP4 is unique. It isn't on the surface.
Where is it?
It is buried inside the capsid, tightly associated with the RNA core. It acts like a locking pin. It solidifies the whole structure.
So it holds the tank together.
Until it touches a cell. This is where the mechanism gets really fascinating. When the virus binds to a receptor on a human cell, the capsid undergoes a massive conformational change. VP4 is ejected.
It literally drops out of the virus.
Yes, it drops out.
Like pulling the pin on a grenade. In a way, yeah. The loss of VP4 loosens the association between the other surface proteins. It opens up a pore or a channel in the capsid vertex, which allows the RNA genome to slip out of the shell and inject directly into the host cell.
Before we get completely inside the cell, I want to touch on something from our supplementary text, Jowett's. It talks about the canyon hypothesis. This seems really important for understanding why our immune system struggles against these things.
This is a beautiful piece of structural biology. If you look at the surface of the virus, at the vertices of the icosahedron, there is a deep depression, a literal canyon.
A groove in the protein shell.
Yes. And the receptor binding site, the actual part of the virus that locks onto the human cell, is located at the very bottom of this canyon.
Why does that matter clinically?
Think about an antibody. An antibody is a relatively large, bulky, Y-shaped protein. It is simply too big to fit into that narrow canyon. So the virus hides its most critical feature, the key to the door, deep in a hole where your antibodies can't easily block it.
That is incredibly devious. So the antibodies can bind to the rim of the canyon, but they might not actually neutralize the virus.
Exactly. The surface loops on the rim can constantly change, that's antigenic drift, without ever affecting the critical binding site at the bottom. However, this structure also gave us a really clever target for drugs.
You're talking about pleconero.
Yes. Scientists developed antiviral drugs designed to be small enough to physically fit into that canyon.
Oh, wow.
The drug binds to the canyon floor and essentially acts as a molecular doorstop. It creates a rigid structure so that even if the virus hits a cell, it cannot undergo that conformational change we talked about.
So VP4 can't drop out.
Exactly. The virus is frozen.
It effectively superglues the tank shut so the payload can't get out. It's a shame those drugs haven't seen wider clinical success due to side effects, but the mechanism is a perfect learning point. Let's talk about that payload now, the genome itself.
This is a classic exam question. Picornaviruses have a positive sense, single-stranded RNA genome.
We hear positive sense a lot. Connect the dots for us. What does that actually mean for the infection cycle?
It means the viral genome is messenger RNA. To the host cell's ribosomes, our protein-making factories, this viral RNA looks exactly like the cell's own instructions.
So it doesn't need to bring any specialized tools with it. It doesn't need to pack a polymerase enzyme in the capsid.
No. It travels extremely light. In fact, there's a concept called the naked genome rule.
Right.
If you take a poliovirus, strip away the protein capsid entirely, and microinject just the purified RNA into a human cell, that cell will get infected and produce new living viruses.
Just the RNA is infectious by itself.
Yes, because the cell reads it immediately and starts making viral proteins.
Yeah.
Compare that to a negative sense virus like measles or rabies. If you injected their naked RNA, nothing would happen. The host cell wouldn't know how to read backwards RNA.
That is violently efficient. Now, the text also mentions a protein called VPG attached to the end of the genome. What is that doing there?
Well, normal human mRNA has a chemical structure at the 5' end called a cap. The ribosome physically looks for this cap to know where to attach and start reading.
Okay.
Picornaviruses don't have a cap. Instead, they have this VPG protein strapped to the end. It helps package the RNA into the capsid and plays a role in initiating RNA synthesis later.
But if they don't have a cap, how do they trick the human ribosome into attaching in the first place?
That is the genius of the IRES, the internal ribosome entry site.
IRES!
It's a complex, highly folded loop of RNA at the beginning of the genome. It basically acts as a physical grappling hook. It grabs the ribosome directly and forces it to start translating. It bypasses the need for the 5' cap entirely.
Which perfectly transitions us into the replication cycle itself. We are moving to section 2 of the Murray outline. The virus has attached, injected its RNA, and the IRES has grabbed a ribosome. What happens next?
The virus pulls a truly ruthless maneuver. It wants to shut down the host's own production lines to eliminate competition. So the very first thing it translates is a protease enzyme called 2A.
The cleaver.
Yes. This protease 2A hunts down a specific host protein called EIF4-G. Now, this host protein is absolutely essential for the cell to translate its own cap mRNA.
Okay.
And the viral protease just slices it in half.
So the cell can no longer read its own instructions.
Correct. The cell's mRNA is effectively silenced. But remember, the virus doesn't use a cap.
It uses the IRES.
Exactly. So the viral RNA can still be translated perfectly fine. It is a complete hostile takeover. The cell stops maintaining itself and devotes 100% of its ribosomes and energy to making viral proteins.
And the product of this translation is weird too, right? It doesn't make separate proteins one by one like a normal gene.
No, the ribosome reads the entire viral genome from start to finish and prints out one gigantic protein chain. We call it a polyprotein.
Just a single massive string containing every part of the virus.
Right. Imagine knitting a sweater, a hat, and a scarf all in one continuous piece of yarn. To make them useful, you have to cut them apart.
Makes sense.
That is what the viral proteases do. Protease 2A and another one called 3C. They chop this giant polyprotein string into the functional units. The RNA polymerase, the VP1 through 4 structural proteins, and more proteases.
And once these parts are assembled into new viruses?
The end game is lysis. The cell bursts open. It is a strictly cytolytic infection. A single cell can release up to 100,000 new viral particles.
And that lysis, that bursting, is what causes the actual damage in the patient.
Exactly. When we talk about polio causing paralysis, it is not secreting a neurotoxin. It's simply that the virus replicated inside the motor neuron and then blew it up to get out. Dead neurons don't fire.
Let's follow that path of destruction. We are moving into the enteroviruses specifically now. Section 3, pathogenesis and immunity. You called it the fecal-oral highway earlier. Walk us through the journey of a poliovirus entering a child.
It starts at the mouth. The virus enters via contaminated hands or water. It first encounters the lymphoid tissue in the pharynx, specifically the tonsils. It replicates there.
Which is why you can sometimes find the virus in throat swabs early on in the infection.
Right. Then it is swallowed. It survives the stomach acid, thanks to that tank-like capsid, and reaches the Peyer's patches in the intestine. It replicates massively there in the gut mucosa.
At this point, is the child sick?
Usually not at all. Maybe a very mild fever, maybe nothing. This is the incubation period. The virus is actively shedding in the stool, making the child highly infectious to others. But the host is relatively okay. But then, the virus crosses the gut wall and enters the bloodstream. This is the primary viremia.
A minor spread.
Yes. The virus circulates and finds the reticuloendothelial cells, the liver, the spleen, and the lymph nodes. It enters these organs and replicates again, severely amplifying its numbers.
It's building an army.
Exactly.
Yeah.
Which leads to a massive release of virus back into the blood. This is the secondary viremia.
And this is the dangerous phase.
Highly dangerous. Now you have a huge titer of virus in the blood, and it can reach its specific target tissue. This is where the concept of tropism comes in.
Tropism meaning the specific cells the virus likes to infect.
Right. All enteroviruses follow this exact highway, but the off-ramp they take at the very end is different. Poliovirus has a tropism for the anterior horn cells of the spinal cord and the brainstem.
And the others.
Coxsackie-B has a tropism for the heart and the pleura. Coxsackie A16 has a tropism for the skin and mucous membranes.
So the disease you get depends entirely on where the virus decides to park after that secondary viremia.
Correct. And this journey gives our immune system two distinct opportunities to stop it. This is absolutely crucial for understanding the vaccines later on.
We are talking about IgA versus IgG.
Yes, secretory IgA lives in the mucosa of the gut and the throat. If you have immunity here, you block the virus at the front door. It can't even establish that initial infection in the Peyer's patches. This prevents infection and shedding entirely.
But if the virus gets past the gut, if you don't have IgA,
then you rely on serum IgG. This is the antibody patrolling the bloodstream. It can't stop the gut infection, but it can intercept the virus during the primary or secondary viremia phase. It neutralizes the virus in the blood before it can ever reach the brain or the heart.
So IgA prevents the infection itself, while IgG prevents the severe disease.
That is a perfect summary to lock in your memory.
Let's talk about the timing of all this. Section 4, epidemiology. When do we actually see these infections in the clinic?
We call it the summer of Entero. Unlike the flu or RSV, which are classic winter respiratory viruses, enteroviruses peak in the summer and early autumn.
Why is that?
It's partly related to hygiene and summer water activities, but also just the virus's environmental stability. In temperate climates, if a child comes into the ER with a fever, an unexplained rash, or viral meningitis in August or September, enterovirus should be at the very top of your differential diagnosis.
And we mentioned they are tough, but how tough are we talking?
They resist standard municipal sewage treatment. They can survive in shellfish that filter contaminated water. And critically, for the summer aspect, they are resistant to standard concentrations of chlorine.
So a poorly maintained public swimming pool can be a major vector.
Absolutely. You need high-level, sustained chlorination to reliably kill them.
Let's get into the clinical meat of this deep dive. Section 5, clinical syndromes. This is what you need to recognize on the wards or your board exams. Let's start with the big one, polio.
Poliovirus. The really tricky thing about polio is the polio pyramid. We tend to only think about the top of the pyramid, which is the paralyzed child, but that outcome is actually rare.
What's the base of the pyramid then?
Asymptomatic infection. Fully 90% of people infected with wild poliovirus have no symptoms at all. The virus replicates in their gut, they shed it, they spread it to the whole neighborhood, but they never even know they had it.
That is terrifying. That is what makes eradication so incredibly hard. You can't see the chains of transmission.
Exactly. The virus moves silently. The next level up the pyramid is abortive poliomyelitis. That's about 5% of cases. This is just a nonspecific febrile illness. Fever, headache, maybe a sore throat. It lasts a few days and resolves completely.
Then we get to non-paralytic polio.
This is aseptic meningitis, about 1 to 2% of infections. The virus reaches the meninges surrounding the brain and spinal cord. The patient gets a stiff neck, back pain, photophobia, but the actual motor neurons are spared. They recover.
And finally, the tip of the pyramid, paralytic polio.
Depending on the specific strain in the population, this is only 0.1 to 2% of total infections. It typically follows a biphasic course.
Meaning 2 phases.
Right. You get the minor illness, you appear to recover for a few days, and then the fever spikes again, accompanied by severe muscle pain and rapidly progressing weakness.
What specifically defines the paralysis? What are the buzzwords for the exam?
It is an asymmetric flaccid paralysis.
Let's unpack that. Asymmetric.
It does not affect both sides of the body equally. It might take out the left leg and the right arm, or just one single leg. This strongly distinguishes it from something like Guillain-Barré syndrome, which is usually ascending and highly symmetric.
And flaccid.
Limp. The lower motor neuron in the anterior horn of the spinal cord is physically destroyed. The muscle loses its innervation entirely and goes completely slack. Over time, because it's not receiving any signals, it atrophies severely.
Is there any sensory loss?
No. And that is a massive key exam point. The sensory nerves travel in the posterior horn of the spinal cord. Polio specifically attacks the anterior horn, so the patient can feel everything, and the muscle spasms are often intensely painful, but they simply cannot move the limb.
The most lethal form of this is bulbar polio, right?
Yes. This happens when the virus attacks the cranial nerve nuclei in the brainstem, the bulb. This region controls swallowing and breathing.
The diaphragm.
Right. Before modern ventilators, this was almost a guaranteed death sentence. This is what the iron lung was invented for, to use negative pressure to physically breathe for patients whose respiratory drive had stopped working.
And briefly, before we move to Coxsackie, what is post-polio syndrome?
This is a real tragedy that occurs 30 or 40 years after the original paralytic illness. Patients who had stabilized suddenly start getting weaker again in the originally affected muscles.
Is the virus reactivating? Like how chickenpox reactivates as shingles?
No, the virus is long gone. The leading physiological theory is neuronal exhaustion.
Or how does that work?
When the original motor neurons died during the infection, the surviving neighboring neurons sprouted new branches to reconnect to the orphaned muscle fibers. They compensated.
Right.
But that means they have been doing the work of 3 or 4 neurons for decades. Eventually, they just burn out and die. It's a mechanical failure of the body's compensation mechanism, not an active infection.
That's fascinating and incredibly sad. Let's move to the coxsackieviruses. These are notoriously confusing for students because the names sound so alike and they cause so many different things. We need a solid mental framework to keep them straight.
The absolute best way is to stick to the 2 main groups, Coxsackie A and Coxsackie B. And I use a mnemonic based purely on the letters.
Let's hear it.
Coxsackie A. Think A for airway and acral meaning the extremities. Or loosely think above the neck and the skin.
What are the key clinical syndromes for A?
First is herpangina. This is a sudden high fever illness with a severe sore throat. If you look in the child's mouth, you see these vesicular ulcers. But here is the critical differentiator from herpes.
What is it?
In herpangina, the ulcers are strictly on the soft palate and the uvula, the posterior pharynx.
And a herpes.
Herpes simplex typically causes gingivostomatitis in the anterior part of the mouth, the gums, the lips, the front of the tongue.
Okay, that's perfect.
So remember, back of the throat equals Coxsackie A. Front of the mouth equals herpes.
That is a solid clinical pearl. And the second syndrome for A?
Hand-foot-mouth disease, usually caused specifically by Coxsackie A16. You get painful vesicles on the palms of the hands, the soles of the feet, and inside the mouth. It is extremely common in childcare settings. It looks very alarming to parents, but it's usually benign and self-limiting.
Okay, now let's map out Coxsackie B.
Think B for body or below the neck, or specifically breast and blood pump. Coxsackie B tends to cause much more systemic internal organ disease,
The classic one is pleurodynia.
also known as Bornholm disease, or, my favorite medical term, the devil's grip.
That's quite a name.
And that name is not an exaggeration. Patients experience sudden, spasmodic, absolutely excruciating chest pain. It attacks the intercostal muscles between the ribs.
It must be terrifying. It probably feels exactly like a heart attack.
It perfectly mimics a myocardial infarction or a pulmonary embolism. But usually the patient is younger, there's a recent viral prodrome, and the pain is highly reproducible with movement or deep breathing.
But Coxsackie B can actually attack the heart too, right?
Yes, myocarditis and pericarditis. Enteroviruses are a leading global cause of viral myocarditis. This can be rapidly fatal, especially in newborns presenting with neonatal sepsis, or it can lead to silent damage that causes dilated cardiomyopathy in adults years later.
Is there a connection to diabetes as well? I've seen that mentioned.
There is a very strong epidemiological association between Coxsackie B infection and the subsequent onset of type 1 diabetes. The working theory is that the virus specifically infects the islets of Langerhans and the pancreas, the beta cells that make insulin, and the resulting damage triggers an autoimmune destruction of those cells.
So to recap for everyone taking notes, A is for airway and a scrawl, herpangina, hand, foot, mouth. B is for body and blood pump.
Yeah.
Pleurodynia, myocarditis.
Exactly. Keep that strict separation and you'll be fine.
Let's touch on meningitis again briefly. If a student sees a case of aseptic meningitis, what should be their first thought?
Enterovirus. Coxsackie or echoviruses cause about 90% of all viral meningitis cases. And remember, aseptic just means the routine bacterial culture comes back negative. There's no pus.
How do we differentiate that from bacterial meningitis definitively? This brings us to section 6, laboratory diagnosis.
You have to do a lumbar puncture and analyze the cerebrospinal fluid, the CSF. You are looking for the classic viral pattern.
Walk us through those metrics.
First, white blood cells. They will be elevated, usually in the range of 10 to maybe 500 cells. In bacterial meningitis, it's usually in the thousands. Also, the specific cell type matters immensely.
Right.
In viral meningitis, you see a predominance of lymphocytes. In bacterial, you see neutrophils.
Second metric, protein.
Normal to slightly elevated in viral. In bacterial, it's usually sky high because of all the heavy bacteria and cellular debris.
And the most important differentiator.
Glucose. This is the absolute deal breaker on an exam. In bacterial meningitis, the bacteria literally eat the glucose in the CSF for energy. So CSF glucose is very low.
And viral.
In viral meningitis, like from an enterovirus, the virus does not metabolize sugar. So the CSF glucose remains completely normal.
So normal glucose equals viral. Low glucose equals bacterial or sometimes fungal or TB. That is the golden rule. How do we confirm the specific virus then?
RT-PCR. It detects the specific viral RNA directly in the CSF. It is incredibly fast, highly sensitive, and has completely replaced the old viral cultures which used to take weeks to grow.
Let's move into section 7, treatment and prevention. This is a massive topic for public health history. The polio vaccines.
The story of the polio vaccines is really a story of a complex trade-off between individual safety and global community efficacy. We have two main tools, IPV, the Salk vaccine, and OPV, the Sabin vaccine.
Let's analyze IPV first, inactivated polio vaccine.
Jonas Salk developed this. It is a formalin-killed virus you inject into the arm. Because it goes directly into the tissue and bloodstream, it stimulates a very strong IgG antibody response in the serum.
So if the wild virus gets into the blood, these IgG antibodies kill it immediately.
Right. It prevents the viremia phase. Therefore, it completely prevents the virus from ever reaching the spinal cord. It protects the individual from paralysis.
But what about the gut?
That is the crucial limitation of IPV. It does not induce significant secretory IgA in the mucosal lining of the gut.
Okay.
So an IPV vaccinated person can still ingest the wild virus. The virus can replicate happily in their intestines, and they can shed live virus in their stool for weeks. They are personally protected from the disease, but they can still be a silent carrier and infect unvaccinated people.
Now contrast that with OPV, the Sabin vaccine.
This is a live, attenuated virus, meaning it's weakened but alive.
Yeah.
You take it orally, usually on a sugar cube or just liquid drops. It perfectly mimics the natural route of infection.
So it goes to the gut.
Yes.
Yeah.
It goes to the gut, replicates in the Powers patches, and induces massive amounts of secretory IgA.
So it creates that firewall at the mucosal surface we talked about?
Exactly. It prevents infection entirely. It completely breaks the chain of community transmission. Plus, because the vaccine virus itself sheds in the stool, it can spread to close contacts and actually vaccinate them indirectly.
Oh, wow.
This is called secondary immunization. It's incredibly powerful for herd immunity in areas with poor sanitation.
So OPV sounds vastly superior for a global eradication campaign. Why don't we use it in the United States anymore?
Because of the inherent risk. Since it is a live virus replicating in the gut, it can mutate. In very rare cases, we're talking about 1 in maybe 2 to 4 million doses, the attenuated virus mutates and reverts back to a fully virulent paralyzing form.
Oh.
This causes VAPT, vaccine-associated paralytic polio.
So the vaccine itself actually causes the disease it's trying to prevent.
Yes. And by the late 1990s in the U.S., because wild polio had been eliminated here, the only cases of paralytic polio we were seeing were caused by the oral vaccine itself.
The math flipped.
Exactly. The risk of the vaccine became demonstrably higher than the risk of catching the wild disease. So the U.S. switched to an all-IPV injection schedule in 2000.
But globally, we still use OPV in the eradication efforts.
Yes. In endemic areas, we still rely heavily on OPV because it is the only way to effectively stop the silent transmission in the gut. The overarching WHO strategy is use OPV to completely corner and stop the spread of the wild virus, and then once wild polio is certified eradicated, switch the whole world to IPV to prevent any further cases of VAPP.
That's a brilliant strategic pivot. Now, are there any actual drugs for treatment once you're infected?
Sadly, no. Pleconerol, that canyon-blocking drug we discussed, failed FDA approval due to side effects and drug interactions. Treatment for all enteroviruses is purely supportive. Iron lungs in the past, modern ventilators today for polio, pain management for pleurodynia.
Let's move to our final major group, Section 8, rhinoviruses. We have covered the tanks extensively. Let's finish up with the drones. Why is the common cold so incredibly common?
Rhinoviruses are the single most frequent cause of the common cold. And we already discussed why they stay strictly in the nose. They love that cool 33-degree temperature, and their capsids fall apart in stomach acid.
But the symptoms, the severe runny nose, the congestion, the headache, is that the virus destroying millions of our nasal cells through lysis?
Surprisingly, no. Unlike poliovirus blowing up motor neurons, rhinovirus doesn't actually cause much cell death or damage in the nasal lining.
Really?
Yeah. The symptoms are almost entirely immunopathology. The presence of the virus triggers our own immune system to release massive amounts of bradykinin and histamine.
So our own aggressive immune response is what actually makes us feel so miserable.
Yes. Bradykinin causes profound vasodilation, which gives you the stuffy, congested nose, and vascular permeability, which causes the runny nose. It also directly stimulates nerve endings, causing the constant sneeze reflex and the sore throat.
Why don't we have a vaccine? I mean, we are on the verge of curing polio, a vastly more severe disease, but we can't cure the sniffles.
It is entirely a numbers game. There are 3 serotypes of poliovirus to put in a vaccine. There are over 100 distinct serotypes of rhinovirus.
Wow.
Generating a vaccine against 1 or 2 simply doesn't protect you against the other 98.
And the natural immunity doesn't last either, does it?
No, it doesn't. Your mucosal IgA protection wanes significantly after about 18 months. And because the virus constantly undergoes antigenic drift, changing its canyon rim, you can easily get infected by the exact same strain a few years later.
Is there any reliable treatment? Does slamming vitamin C actually work?
Not really. High-dose zinc lozenges have some mild clinical data suggesting they might physically block the viral ICAM-1 receptor if taken very early in the symptom course. But mostly it's about symptom management, vasoconstrictors, and ironically, chicken soup.
Does the heat actually help?
Theoretically, yes. The steam from the soup creates an environment in the nasal passages that is too hot for the virus's optimal 33-degree replication, while simultaneously hydrating the mucosa and thinning the mucus. It's surprisingly evidence-based comfort care.
That is fantastic. We have covered a massive amount of ground today, from the naked capsid structure all the way to the depths of the polio pyramid. Before we get to the rapid recall quiz, what is your big picture takeaway for the listeners?
The Picornaviruses perfectly illustrate that genome size does not determine clinical impact. A tiny, simple strand of positive sense RNA, by simply varying its protein code stability acid stable versus labile and its receptor preference, can choose to be a minor winter nuisance or a lethal, paralyzing killer, It's a masterclass in viral efficiency.
And we are seeing nature continuously fill the void. As wild polio disappears thanks to the vaccines, we are increasingly seeing outbreaks of non-polio enteroviruses, like Enterovirus D68, causing acute flaccid myelitis, essentially paralysis in kids. The biological niche is still there.
Absolutely. We might be eradicating polio, but the enterovirus family is far from finished. Are we just trading one paralyzed nervous system for another? The non-polio enteroviruses are definitely the next major frontier in neurology. We need to stay highly vigilant.
That is a sobering thought to leave on. All right, let's test what's stuck for everyone listening. It's time for the rapid recall quiz.
I love this part.
Listeners, I will read a short clinical vignette or question. I'll pause to let you think of the answer, and then we will give the correct answer, and most importantly, the why.
Let's do it.
Question 1. A 10-year-old boy presents to the emergency department with severe chest pain that comes in sudden spasms. He says it, quote, hurts to breathe. His ECG is completely normal. He had a mild fever a week ago. Which specific subgroup of enterovirus is the most likely cause? Take a second to think about that. The answer is coxsackie-B.
And why? This is the classic presentation of pleurodynia, or Bornholm disease. Remember the mnemonic. Coxsackie-B is for body or breast chest. Coxsackie-A is usually pharyngeal or skin-related. If you see chest pain with a normal ECG in a young person, think coxsackie-B.
Question 2. You are analyzing the cerebrospinal fluid of a patient with suspected viral meningitis. What glucose level do you expect to see? High, low, or normal? Take a second. The answer is normal.
Why? Viruses do not consume glucose for metabolism. If the CSF glucose is abnormally low, hypoglycemia, you must immediately suspect bacterial, fungal, or tubercular meningitis, as those organisms eat the sugar. Normal glucose points directly to a viral etiology like enterovirus.
Question 3. Which polio vaccine is capable of causing contact immunity or secondary immunization in unvaccinated people? Take a second. The answer is OPV, the oral polio vaccine or Sabin vaccine.
Why? OPV is a live attenuated virus that actively replicates in the gut mucosa and is heavily shed in the stool. This fecal shedding can infect and therefore immunize close contacts. IPV is a killed virus injected into the arm and is never shed.
Question 4. A 4-year-old child presents with a high fever and a severe sore throat. On physical exam, you see vesicular lesions exclusively on the soft palate and the uvula. The lips and the anterior gums are completely clear. Is this herpingina or herpes simplex? Take a second. The answer is herpingina, caused by coxsackie A.
Why? Herpingina affects the posterior pharynx, so the soft palate and uvula. Herpes simplex gingivitis typically affects the anterior mouth, primarily the gums, lips, and hard palate. Location is everything for this differentiator.
Question 5. Structurally speaking, why are enteroviruses highly resistant to stomach acid while rhinoviruses are rapidly destroyed by it? Take a second. The answer is intrinsic capsid stability.
Why? Both are naked, non-enveloped capsids, but enteroviruses have evolved a tightly bound protein structure that remains perfectly stable at a pH of 3. Rhinoviruses are acid-ligolal. Their capsid undergoes a fatal conformational change and falls apart in acidic environments.
Question 6. What is the specific function of the IRES in the Picornavirus genome? Take a second. The answer is to initiate translation without needing a 5' cap.
Why? The viral RNA does not have the standard 5' methylguanosine cap that normal human mRNA uses to attract ribosomes. The internal ribosome entry site is a folded RNA loop that allows the virus to physically hijack the host ribosome directly, bypassing the host's normal translation rules.
Question 7. Which viral protein is buried deep inside the capsid and acts as the locking pin that is ejected upon receptor binding? Take a second. The answer is VP4.
Why? VP1, VP2, and VP3 form the tough outer shell of the icosahedron. VP4 is strictly internal and is ejected to loosen the capsid and allow the formation of the pore for RNA injection into the cell.
And that brings us to the end of Murray Chapter 46.
If you keep those tank versus drone distinctions clear in your mind and remember the A versus B mnemonics, you will absolutely ace this section on your boards.
Join us next time as we continue our journey through the microscopic world. Safe travels on the fecal oral highway, everyone.
And please, wash your hands.